So, hi everybody. We are going to start our second part of the special senses, continuing with the sense of vision. So we stopped last time talking about the medical conditions for what we better known as nearsightedness and farsightedness and astigmatism. Now we are going to dive in a little bit deeper and talk about how your eye actually creates vision using the third layer of the eye, which is identified as retina or your sensory tunic. So first of all, the sensory tunic itself is made up of two layers. It has an outer layer, which is identified as the pigmented layer. This layer is actually very similar. If you look at it, it's very similar to the choroid layer we discussed last time. Remember, choroid layer contained a significant amount of melanin, the color pigment, and that allows us to prevent the light from scattering within your eye and basically limit your ability to create a vivid image. Pigmented layer also is the location for storage of vitamin A that is necessary for your vision. Now, the second layer of the eye, which is the innermost layer of the eye, it's the lining of the internal aspect of the eye, is referred to as the neural layer. Again, kind of look at the word neural. This implies neurons that are going to be three different types: photoreceptors, bipolars, and ganglion cells. Photoreceptors are identified as modified neurons with the goal of creating images either in the presence of light or in the absence of light. Now notice again, there are two types. One type are called cones. These are the ones that are going to work when you are in a bright environment, and it's going to give you, again, a vivid image, high quality vision. And in reality, there are three different types of photoreceptors for cones that are sensitive to blue, red, and green. So basically, any color that you see is a result of the light activating different combination of these blue, red, or green cones, giving you, again, range of colors and variation in colors. On the other hand, when we talk about your rods, which are the second type of photoreceptors, these type of cells are the ones that are basically give you the capacity to see in a dark or dim environment. Kind of imagine if you're walking to your house and your house is dark, you may be able to see where your furnitures are. You can see kind of the shades and the areas where the items are located, but it doesn't really give you any details or vivid image. So the goal of rods is basically for you to create a vision in dim or dark environment, and therefore it's going to be extremely sensitive to light. Understand that rods are sensitive to light because you only need very small amount of light to get them activated. Cones, on the other hand, are considered to be least sensitive to light because, again, you need a lot of light. You need bright light environment to cause your vision to be created. So they're considered to be least sensitive. Now, when the image is created by your photoreceptors and how that image is created, which is a process called phototransduction, I'm not going to touch upon it. We'll discuss this in much more detail in physiology. But let's say magically your image was created by your photoreceptors. Now these photoreceptors need to pass the message to another set of cells that are referred to as the bipolar cells. Regardless of bipolar cells. First of all, understand the terminology. Bipolar was a category of the neuron that has two extensions coming off of the cell body. And their goal is to take the signal that your photoreceptors have created, the first type of cells, and move them on across this path that eventually ends in your brain. So bipolar cells receive the electrical impulses from your photoreceptors, which is basically a signal that your photoreceptors have created. and take that message and pass it to the next category of cells, which are ganglion cells. So ganglion cells receive the electrical impulse from the bipolar cells, or receive the signaling, maybe I should say, from the bipolar cells. And these ganglion cells, again, are neurons. Therefore, they will have axons as part of their structures. When you take all the ganglion cells' axons, if you take all the ganglion's axons together and bring them out from the back of the eye, you form the structure known as the optic nerve. And just to kind of refresh your mind, optic nerve eventually ends in the occipital lobe of your brain or your cerebral cortex as your primary visual cortex, where you receive the signals and eventually get that signal processed to give you your sense of vision. So again, kind of looking at this picture, giving you an idea of what this structure looks like. So these cells we have in the back, you can see two colors, purple and yellow. The purple, as you can see, more frequent. These are your rods, and the cones are depicted in yellow. These cells are the ones that are, again, there's 3 sensitivity to red, blue, and green colors. The cells, notice how they are connected right here to the second category cells you see right here in red. These will be identified as your bipolar cells. And what you see in white at the end, these would be your ganglion cells. Now notice how these ganglion cells have these axons coming off of them, and see how they're all merging together. So these merging of these axons, all of these coming together, these would be coming out of the back of the eye, and they will form what we identified again as your optic nerve. The information that's written here is pretty much the same thing we discussed earlier, but you're welcome to review it again. Rods are least sensitive to light. There is no color or sharp image. They're more frequent, and they give you the image dim light, and they are peripheral vision as well, so the more side view that you have. Cones, on the other hand, they require significant amount of light. There are 3 different colors, and they are going to give you high resolution pictures. Here is the same, again, same information, just in a chart format. You're welcome to review these on your own, and let me know if you have any questions. Now, a couple of information about the retina. Now, if you think about the photoreceptors, which are, again, rods and cones responsible for creation of your image, there is a location on the back of the eye, which you can see it highlighted right here, called fovea centralis. Fovea centralis is a region where you will have the highest concentrations of cones, which basically would give you your best visual acuity. So normal conditions where you see the images that are right in front of you, that is where the light is focusing on your fovea centralis, and again, giving you the best image possible. The other structure that is visible on your photoreceptors, or your, more specifically, I should say, on your sensory tunic or your retina, is this area right here, which is identified as the optic disc. Please do not mistake this with optic nerve. So let's take a look at this. The extension that's coming out of the back of the eye, that's your optic nerve, which is the collections of your axons of the ganglions. Now, the area where the axon of these ganglions are actually leaving to form the optic nerve, so this region where the nerves are actually leaving from the back of the eye, that lacks any type of photoreceptors, that is referred to as the blind spot. This is also where you get your blood vessels into your eye for the purpose of basically supplying your eye with oxygen and nutrients. Again, this is the same picture we described before, 3 layers of cells. You have your rods, you have your bipolar cells and the ganglion cells, and the axon of the ganglion cells are what eventually become your optic nerve. So if I ask you guys, tell me the pathway for the signaling traveling through your, or signaling that gives you your ability to see. Basically, you start again with photoreceptors. The job of your photoreceptors, simply put, in a general terms, regardless if they're rods or cones, is to take light, right? Take light, photoreceptors are sensitive to light, so take light and convert that to an electrical impulse. Now that signal gets passed on to your bipolar cells, eventually to your ganglion cells. The axon of the ganglion cells come out as optic nerve. Optic nerves are going to crisscross on a structure called optic chiasm. So kind of imagining again, if these are your two eyes, the extensions that are coming out of the back of the eye, those are your optic nerve, and the area where that kind of crossing happens, that's your optic chiasm. Do understand that this crossing happens because of the whole concept of contralateral, as in right sensory data gets sent to the left and vice versa. Now the continuation of the optic nerve, what you see right here, is referred to as the optic tract, and that eventually ends in the back of the brain, which is right here, called your visual cortex, located in the occipital lobe. Now, a medical condition that is associated with your sensory tunic is referred to as detached retina. So you may have heard of this. A typical sign of somebody having a detached retina is when they start to see a lot of kind of bright spots throughout their vision. That's typically a sign of the beginning of the retinal detachment. So what happens is, if you think about the sensory tunic as a whole, it has two layers. We mentioned that earlier. One layer was the pigmented layer, and the other one is the neural layer, which is basically your photoreceptors. So neural layer, pigmented layer. This is the one that is dark, that contains melanin, and neural layer is what contains your photoreceptors. Now, if these two layers separate from each other, that's the detachment. The problem is that your photoreceptors have no access to any blood supply, because the blood supplies come from the vascular layer, and at that point your cells will start to die. If it's not treated, it can lead to permanent blindness, and if it happens suddenly, it could happen as a result of some sort of head trauma or sudden stopping of the head during some movement, a typical example being a bungee jumping. And again, this sort of trauma can literally rip these two layers apart from each other and lead to the death of your photoreceptors. Another type of condition you see associated with your photoreceptors is atypical blindness, or color blindness, sorry. It is inability to see certain colors or distinguish certain colors. The main form of color blindness is inability to distinguish the color red from green. So you can see on the left-hand side, I have a circle. There is a number 8 written in red in a background of green. So if you have a hard time seeing the red color from the background, that means the red and green photoreceptors are not working properly. It might be other colors. However, interesting fact about this is if you are in a very bright environment, you may be able to distinguish these colors because you activate enough of those green and red photoreceptors to make that distinction. However, in artificial light, like say you're in a room, a fluorescent light, these make it very difficult to separate. And sometimes the object appear more of a brown color than the green and red we expect to see. Now, if we take a look at your eyes, we have two main breakdown or segmentation within the eye, what we identify as a posterior segment and what we identify as anterior segment. So let me show you guys in the picture first. So if you imagine here is your eye, this is a space that is grayed out. So this space right here, that's the posterior. And what is depicted here, from here to here, is your anterior segment. So anterior segment versus the posterior segment. Now, what is the posterior segment contains? What does posterior segment contain? It contains a fluid referred to as the vitreous humor. If you ever get a chance to dissect a cow eye, if you break into the posterior segment, you can see this kind of a gelatinous material that comes out, and that's basically the vitreous humor. Main function, it transmits lights as the light passes through the eye. It allows the two layers of the neural layer to hold against the pigmented layer. So again, you want these two layers to be directly connected so your neural layer can receive its nutrients and oxygen. It does also contribute, this fluid contributes to the intraocular pressure, which is extremely important. And it's the source of this vitreous humor is coming from the embryo and basically lasts your whole life. Anterior segment, again, front portion, is actually divided to two chambers, anterior chamber and posterior chamber. Now, anterior chamber, as in front of the front, if you want to think about it, within the anterior segment, we have an anterior chamber. That's between the cornea and the iris. And the posterior chamber of the anterior segment is between the iris and the lens. So let's take a look. Again, we identified this whole structure as the anterior segment. Now, this region right here, again, between the cornea and your lens, which is right here, this is referred to as the anterior chamber. And this space right here will be identified as the posterior chamber. Both of these together is this breakdown right here, which is the anterior segment. Now, what is the fluid? The fluid is called aqueous humor, and it's a plasma-like fluid, so it's more watery, as the name aqueous implies, and it is continuously formed by the capillary of the ciliary process. So let's show it to you. So here is basically your ciliary process right here, these holes. So fluid basically enters into the posterior chamber. It works its way up through the pupil into the anterior chamber, and then eventually gets picked up with these holes inside your fibrous tunic, which are referred to as the scleral venous sinus. Literally, that fluid gets picked up. So this fluid is continuously being produced, coming through the two chambers, and then eventually draining into your scleral venous sinus. So that is all I need you guys to know about your sense of vision. Now we are going to talk about your ear and equilibrium. However, I'm going to stop the lecture here. It's on the shorter side, and then we're going to do your ear and equilibrium for the third part of the special senses.